Hello everyone, my name is Alex Gaspar, and thank you for joining our research symposium this afternoon. Today, I will be discussing our research into the design, synthesis, and biological evaluation of Donepezil-based acetylcholinesterase inhibitors as therapeutics for Alzheimer's disease. Now, this great research was made possible by our awesome Principal Investigator, Dr. Stevan Pecic, at California State University of Fullerton's Department of Chemistry and Biochemistry. So, what exactly is Alzheimer's disease and why should we care about it? Alzheimer's disease is a deadly and debilitating neurodegenerative disorder that is characterized by significant impairments in cognitive functioning, memory, and learning. Millions of people are affected by it worldwide. And if you take a look at some of these scary statistics, you'll see that Alzheimer's disease is increasing at an alarming rate. Not only that, but along with other forms of dementia, it's one of the leading causes of death in our senior citizens. Lastly, it takes a huge economic toll on our country. Therefore, it's very important for medicinal chemists to research, design, and create drugs that will serve as therapeutics for Alzheimer's disease. Although Alzheimer's disease is currently incurable, there are some FDA-approved drugs that seek to treat the symptoms of Alzheimer's disease. One key symptom of Alzheimer's disease is decline in cognitive functioning, memory, and learning. And one drug that treats this is known as Donepezil. It belongs to a class of drugs known as acetylcholinesterase inhibitors. Now, if you take a look at the graphical abstract at the top right, you can see how acetylcholinesterase is an enzyme that breaks down the neurotransmitter acetylcholine. If this neurotransmitter is being broken down, then this neurotransmitter can't help facilitate memory and learning between our neurons. These are areas of concern for Alzheimer's patients, so Donepezil is very helpful in inhibiting this enzyme and allowing memory and learning to improve in Alzheimer's patients. The focus of our research is to design better Donepezil analogs that can be more potent and have better inhibitory effects to have even more potential benefits for Alzheimer's patients. Our first step in finding new and improved acetylcholinesterase inhibitors was to first design 4 Donepezil-based analogs and visualizing exactly how they would fit in the binding pocket of acetylcholinesterase. We did this by first seeing exactly how Donepezil was fitting into acetylcholinesterase. And we saw that it was fitting really well, but there was some room where we could add some bulkiness and different structures to improve its attachment into acetylcholinesterase. Afterwards, we synthesized our 4 analogs using commercially available chemicals in microwave synthesizer. The microwave synthesizer left us with a crude product containing our desired compound with some extra byproducts. We purified this using flash chromatography in a Combi Flash RF system. Once we had our purified inhibitors, we biologically evaluated them using Elman's method. Elman's method was a great way to measure inhibition potency by seeing exactly how much of our inhibitor was working as intended. Acetylcholinesterase from an electric eel and acetylthiocholine, which was a derivative of acetylcholine, was used in each of these microplate assays as well as our inhibitors. As you can see, the yellow color indicated that no inhibition was taking place, while a lighter color or no color meant inhibition was taking place. We then used a microplate reader to measure the half-maximal inhibitor concentrations of all 4 of our inhibitors, along with Donepezil for comparison. These values are also known as IC50 values, and they're measured in nanomolar concentrations. They give us a great idea of how potent our inhibitors are at inhibiting acetylcholinesterase. What we found was that inhibitor 1, inhibitor 3, and inhibitor 4 were not significant inhibitors for our study, but inhibitor 2 was a really potent inhibitor of acetylcholinesterase. So what exactly does all of this mean? Well, it's amazing. We were able to design a drug analog that showed potent inhibition of acetylcholinesterase. In fact, what we did was replace the benzene ring of donepezil with an isoquinoline group as boxed in purple above. And now what we can do is open up new doors to future drug analogs by placing functional groups at the green arrows of this inhibitor 2. This will serve as our new lead compound into the future endeavors of our research into Alzheimer's disease therapeutics. Thank you very much. I would like to acknowledge Project RAISE and the support of the U.S. Department of Education and the support of all of the fellow Pecic Lab members. Thank you very much.